All right. So these gene mutations, again, as I ended the last lecture talking about, are where we are dealing with a substitution, addition, or removal of a nucleotide. But the gene / point, it's called both. You know how we love to make two names for the same thing in biology. These mutations are where we're dealing with only one nucleotide, or more than one, but the nucleotide sequence, the bases, okay? Remember with the chromosomes, we were dealing with entire segments of the DNA sequence moving, whereas here we're dealing with, it's at a more of a micro level with the nucleotides and what's going on. All right, so substitution. I like gene mutations because they've named them for exactly what is happening. And if you just kind of use some common sense, it starts to make sense. A substitution is where we substitute one nucleotide for, like in this case, this is supposed to be G. Instead, because remember, G and C always go together, instead we have an A because this is a T, and so A will bind with T. So sometimes a substitution doesn't affect the amino acid that is coded for it, because if you remember, when we did codon bingo, I think we did codon bingo. I don't know if we ever got it. I don't know if we did with your class yet. But if you remember if you look at your amino acid chart and you look at your sequence of 3 that codes for the amino acids, you can have the letters, different letter combinations code for the same amino acid. But in this case, it changes the amino acid. And it also depends on how different the amino acid that was coded for, how much it changes. Remember, we can group amino acids into families, being basic or acidic or hydrophobic or hydrophilic. So if they're similar, the protein's going to fold similar. If they're totally different, we're going to really affect the protein folding, which thereby affects the protein function. Now, so here we have a substitution, and all it is, going back to that, is one sequence, one nucleotide changing. And again, this could be very inconsequential, or it can cause a disease depending on where the substitution occurs. And what amino acid gets changed. Frame shift, I don't have a demonstration here on the PowerPoint, but that's where if nucleotides are deleted, why we call it a frame shift is it means that the entire way that we would read the groups of 3 changes. And now it's going to change. The problem with the frame shift is it's going to change everything downstream of the frame shift. And so we're going to code for all different amino acids. And this insertion is an example of what would cause a frame shift, because you would add a nucleotide, and it can be one or more nucleotides. And now we added a nucleotide here. If you look, it was supposed to be CTT. We added another C, so we have CCT. Well, now we're changing, this would be the normal one. Now we're changing all the 3 groupings downstream of where you had the insertion. So this is written a little weird in the notes, but insertion and deletion mutations will cause a frame shift mutation. So here we are where we have substitution, one nucleotide replaces another. Frame shift, this would be if nucleotides are deleted, and an insertion mutation is one or more nucleotides added, and it can also cause a frame shift. So which type of gene mutation do you think would cause the most serious errors? Anything that's going to cause a frame shift is going to, now that's a hard question because substitution might cause a serious problem depending on where it is in the protein folding and what it was, where, how it coded. But the most damage collectively we're going to see is any time that there's a frame shift that occurs. Now, point mutation is just the word for both of these, so that one doesn't work. So an example of a substitution error is sickle cell anemia, and that's where I say, frame shift errors are, substitution errors they can cause a disease. It's not to say they're harmless. And this is a great example where it's amazing though, out of how many nucleotides we have in our DNA sequence, that just one could cause a disease. But here's the DNA, and here is the normal chromosome 11, where it calls for glutamic acid after this part of the DNA is transcribed. And here we see where the substitution, it should be GAG. Instead, we have GTG. So what's coded for here is GAC. So it just flip-flopped, right? And now it should have been, the mRNA should have been GAG, which translates into glutamic acid. Instead, we have GUG, which, if we translated that and used our coding chart, we would get valine instead. So one letter caused us to get the wrong amino acid, and the result is we have a sickle-shaped cell or a sickle-shaped red blood cell, okay? And this is an issue because this makes it where the hemoglobin that is sickle-shaped isn't round, and it can't fit into the red blood cells. It'll start to clot and kind of stiffen in the capillaries. This is the normal protein folding that's supposed to happen with normal hemoglobin. The red blood cells will go into like this nice pretty shape. Instead, with the sickle cells, they cannot. It seems like it to be a tongue twister for me. So you need to understand that sickle cell anemia is a result of a substitution error. And you need to know that for your test. That's why we have it underlined and bolded. This is just talking, and at this point, after you did your worksheet and that little movie that you watched about the sickle cell disease, this should make a little more sense. That a normal person has 2 genes, one from each parent, right? And you get normal hemoglobin. However, a person with the sickle cell trait inherits, basically, if they have the trait, they're going to have 1 normal and 1 defective gene. Now, that is where if you did that worksheet, you realize that's actually a good thing, because that means they have some immunity to malaria. But if a person has both